Okay, today I will not talk about pets or any other eschatological issues as yesterday because Didier did it today, so it was more than enough. So I will focus on what we are doing in tricho farming, because in tricho farming we are using this 360 degrees approach in order to elucidate the different properties of Artemisia annua and other Artemisia species, but not only for animal health or human health, but also for plant health and plant protection. But this is the subject of today. Okay, so you know that plant diseases is a big problem. So indeed, pests and weeds cause among the 20 to 40% of losses in the yield in all the crops. From all the different pests, fungi is a very big problem. Indeed, during the cultivation process, is responsible of 10 to 23% of the losses, and during the post-harvesting, which is also a very important issue, from 10 to 20%. So the situation is not that good, but even could be worse. Why? Because of two different important issues. One is the population that is growing. In the last 90 years, the population has increased 4 times, so we are reaching more than 8,000 million people. By 2050, will be 9 million people. So we need, we are a lot of people, and we need food, quality food for everybody. The second bad thing is the climate change. So climate change is affecting everybody. So you can see here the typical fungal disease in the grapes. I'm coming from a region in Spain that is called La Mancha. La Mancha is considered to be the biggest region producing wine in the world. Okay? I'm telling the biggest. I'm not telling the best wine, okay? Because I know that there are a lot of Italian people, a lot of French people, and in minority, so I will not say the best, but we have a lot of wine. So which is the problem now with the climate change? That is, we have hotter temperatures and the rain is heavy, so is this humidity with high temperatures is the best conditions for the fungi to grow. So in the case of the grapes, we have a lot of botrytis or mildew, for instance. So we go to the next. Okay, that is here. No. Okay, yes, I will summarize. So yes, in case, so now there are a lot of chemical fungicides that they have a lot of problems, toxicology, also health issues, and also there are consequences in the soil. And you know that for agriculture, we were speaking yesterday about microbiome. There is a particular microbiome surrounding the roots of the crops that it is important to keep it healthy. So chemical synthesized pesticides is really destroying also the microbiome on the soil. So the alternative that we have is, natural fungicides. And this is what we are talking today. So we go to the next one. Okay, there are two different kinds of natural fungicides, microbial and botanical fungicides. Which is the main problem with the fungicides? That the regulation and the registration process is really, really expensive. For instance, in Europe, you need to spend, just for the registration and to prove that it's working, from 3 to 5 million euros. So imagine who can afford to do that. So nobody, small companies or nobody. Okay, so as you know, botanical extract, all of you know, medicinal and aromatic plants arise as the most important, probably, crops in order to find the properties as biocides. Why? Because we know that, already has been identified more than 50,000 secondary metabolites that are present in the medicinal and aromatic plants, but the prediction that there are among 200,000 to 1 million, secondary metabolites that a few of them, but even if a few of them has property to be antifungal, there is a lot of them because we can reach 1 million of different secondary metabolites. So, okay, all of you know, so the purpose of the research, okay, I will explain you. We not only use the superstar Artemisia, as Didier said, Artemisia afra and Artemisia annua during the project that I will present today, and I would like to highlight that is just a preliminary screening project. We have used 5 different species of Artemisia in order to elucidate their potential of antifungi, okay? Next. Yeah, so You know that plant hairs, trichomes are very, very important. So it's a very important way that the plants they use to defend against themselves. So it's something that we need to take into account. Okay, secondary metabolites, they are very important. Many secondary metabolites, not all of them, but many secondary metabolites, because they could be toxic. So that's why they have medicinal properties, are produced in the glandular trichomes. But also it's important to highlight that plants, medicinal plants, has not only glandular trichomes that are synthesizing, for instance, artemisinin, but has non-glandular trichomes that act as physical barrier to protect the plants against different pests. Indeed, you can see here, this we call the dragon against malaria, because it's just a microscopic picture that I took during my postdoc. And you can see here is just the tip of a leaf of Artemisia annua, okay? And the things that you see on orange is not the glandular trichomes that produce the artemisinin, but are the non-glandular trichomes that are defending the plant against other, other issues. So it's important to take into account, if we go to the next one, that trichomes are very important because they don't not only produce, in the case of Artemisia annua, artemisinin, but they are able to synthesize and to storage other secondary metabolites that I will not go into detail due to the lack of time. But I would like to highlight that most of the secondary metabolites we are using for human health and animal health as immune modulators. And I would like to, maybe some controversy, but I would like to say that, okay, plants also they have their own immune system. Okay, they don't have the typical Th1, Th2 immune system with the T cells, B cells, and cytokinins. But plants, they have their own immune system. They produce a lot of molecules. They use the trichomes to defend themselves. So even these secondary metabolites that are produced in the Artemisia species could be used to increase the immune system of the plants to protect against other pests. So this is important to highlight. Plants has also a particular immune system. Okay, so we pass the next one. Okay, so but this is important, as we say, we need to standardize the content in secondary metabolites in the plant, within the plant, because otherwise we cannot reply all the time the results. So in trichofarming, long time ago, we produced a biostimulant that we call trichostimulant, that we are not able to increase the immune system of the Artemisia annua plant, so increasing the density of the trichomes, therefore increasing the concentration of artemisinin, but also we are increasing the leaf biomass, and most important, we are accelerating the growth of the plant. So we are able to have 3 or 4 crops per year of Artemisia annua. So not only with our biostimulant, that our biostimulant is done by plant extract only. So we are not only increasing artemisinin, but we are increasing flavonoids such as casticin, eupaterin, or quercetin. Okay, we go to the next. So the idea of this project was to have Artemisia annua species treated with our biostimulant, so with a higher concentration of secondary metabolites, and to test their effectivity against the 3 main fungi diseases in plants: Alternaria, Botrytis, and Fusarium. So if we go to the next. Okay, as I told you before, this study was a really preliminary screening. So we wanted to test in vitro the efficiency of this water extract of Artemisia. I would like to highlight that, okay, there is already some publications regarding, the antifungal properties of essential oil of Artemisia, but in this case, we just did water extract, okay? Well, here we are in the same mood. So in order to know that, okay, we have more like polar molecules present in the water extract. Okay, so we go, I will present very briefly the results. The first species that we try is Artemisia absinthium. And as you can see here, we calculate the inhibition. I will not go into detail into technical issues because we don't have time, but we are measuring the inhibition after the treatment with the aqueous extract of Artemisia absinthium. Here you have Botrytis, Fusarium, and Alternaria. As you can see here, unfortunately, there was not a significant inhibitory effect obtained. That means that, okay, so far as Artemisia absinthium should be used to produce vermouth or absinthe, that's far, and it's enough, okay. We are happy for that. We go to the next slide. Artemisia afra. Okay, here we could see that there was a very significant inhibition of the 50% of Artemisia annua water extract regarding botrytis, which was very important. Regarding Alternaria and Fusarium, it was not really, the decrease was not that big and not significant, but okay, there was a significant inhibition regarding botrytis, which was very good. If we pass to the other superstar of Artemisia, Artemisia annua, in this case, there was a strong inhibition as well in botrytis, a higher inhibition regarding Fusarium, as well as Alternaria. So that means that, you know, there was kind of like a higher spectrum of inhibition of different fungi. I forgot to tell before that, before all the extracts, how we prepare was 50 grams of leaves in 1 liter of water, okay? So all the results we present, they were that way. In the case of Artemisia annua, also we tried to go down, okay? We used 25 grams per liter and 12 grams per liter, but you can see that the results, they were a bit lower compared with the 50 grams, but still they were good. But it is clear that there is a correlation between the concentration of the plant and the inhibitory effects of the fungi. But the results, they're fine. If we go to Artemisia dracunculus, which is a tarragon, you know, in the Mediterranean area, we are using like a species to cook. The results, they are really, really good, because when we use 50 grams per liter, we are able to decrease the botrytis almost 80%, which is really, really, really good. Unfortunately, it's not everything perfect. So in the case of Fusarium, there was no results. And in the case of Alternaria, okay, more or less. Here, and similar as we did with Artemisia annua, if we go to the next slide, we decrease also the concentration, but here you can see there was not a change at all. So either using 50 grams, 25 grams, or 12.5 grams, the results against botrytis, they were really, really good. And the rate of inhibition, there was 80%. And last but not least, I would like to present Artemisia tuscula, that I don't know if you have heard about that. Artemisia tuscula before was called Artemisia canariensis, and is an endemic plant used in Canary Islands, okay? There. So now the official name is Artemisia tuscula. Like our crops and our research regarding medicinal plants, we are doing it in Tenerife, in the Canary Islands. We say, okay, let's use this plant, okay? This plant is very interesting because in the local traditional medicine, you know, Canary Islands, they are very isolated islands. So now there are a lot of flight connections, but 1 century ago, nobody was going there, no? So they have to use a lot of, you know, of local medicine. It was very interesting because they were using Artemisia tuscula to make some fences, and they realized that when they put some fences around the cattle or the livestock they was not getting sick, okay? I have to say that we don't know which was the result because the smell of Artemisia tuscula is disgusting. So probably was it more a repellent issue. So if you compare Artemisia afra, Artemisia annua, they are really good smell. Artemisia tuscula, you touch it, it's not for a tea. I would not really recommend it, okay? But okay, we did also, like we are in Tenerife, we say, okay, let's analyze the results. So if you go previously, sorry. Okay, so the results, they were not as good as Dracunculus and Annua, but they're not bad. Okay, there was a significant inhibition of the 3 main fungi. So we need to explore more. If we go to the final slide. Yeah, so just this is a heat map with all the results that I will not in detail, but what we can find that tarragon or Artemisia dracunculus has the strongest inhibition regarding botrytis. Artemisia annua seems to have a broad spectrum against the 3 fungi, and Artemisia tuscula, okay, I'm very happy, I'm very proud of this very, you know, smelly Artemisia that is present in Tenerife. So, which are the future perspectives? Of course, there was a preliminary study. We don't have what is in the water extract, okay? Because, you know, probably there are some polar components that could be like phenolic acids, like, you know, like caffeic acid, chlorogenic acid, or maybe, you know, there is another molecules that we should research in order to know why even the water extract of Artemisia has a high level of antifungal properties. Okay, so what is next? So probably we'll use untargeted metabolomics in order to know what is in there. So in order to explore more in detail, and of course to do in vivo test, which is the most important part. So you're ready for the lunch.
Thank you. Thank you, Luis. Thank you very much.
I have to say this is our Artemisia annua crops in Tenerife. That's
Very
amazing.
beautiful. Thank you very much.